Спасибо. Первое, я приветствую всех. Для меня это большая честь быть участником этой конференции. И я бы приветствовал профессора Трапезова на приглашение. Но сейчас буду говорить по-английски. Но мой английский тоже не является идеальным, но тем не менее. Следующий раз мой доклад будет на русском. Через 2 года, да. I'm Janko Skok from Slovenia, University of Maribor, and today I'm going to present actually 2 topics. First is the parasite mediated domestication hypothesis, and one preliminary study, the first one in the frame of this hypothesis we did recently. So this study was done also in collaboration with other institutions from Croatia and Slovenia. But let me first explain on quick some background. As you probably know, there is a set of phenotypic traits which are common to literally all domestic animals. That traits were mentioned already before. So floppy ears, upturned tail, depigmentation, and so on. And it is termed, this phenomenon is termed domestication syndrome. And as was said already, so domestication syndrome was first explained, described actually by the Darwin, but not explained until more than 100 years later by Belyaev here in Novosibirsk. As you probably know and are familiar with this, so Belyaev in 50s, he started proto-domestication experiment on silver foxes. And as far as I know, it is still ongoing right now, this experiment. So actually, and the main premise he derived from was that the tameability is a key trait, is like an essential precondition of domestication syndrome. He simply said, if you select animals solely on this behavioral trait, tameness, so the frequency of the other domestication syndrome traits will increase in the population. So, and in this way of selection, he created, let's say, 2 types of foxes. So domesticated and aggressive, so tame and aggressive, fearless animals. Okay, here are some results of his experiment, so that proved his expectations, his hypothesis. And on the basis of that experiments, he concluded, he interpreted that results as that selection for solely tameability will destabilize the regulatory systems. For example, HPA axis, this is hypothalamic, pituitary, adrenal axis, that control morphological and behavioral development, which results in changing that are otherwise characteristic for domestication syndrome. So, and later, there were also some other explanation of the mechanisms behind the domestication syndrome. For example, Crockford and her thyroid gland hypothesis. So she claimed that that in domestic animals, the rhythm of thyroid hormone secretion is somehow deviated in the domestic animals, and thyroid glands is it's actually responsible for and that changes are responsible also for heterochronic changes, which were mentioned before. For example, neoteny, pedomorphism, and so on. And recently, Wilkins and his colleagues proposed the neural crest cells hypothesis. So, neural crest cells that are cells appeared in early embryonic phase, emerged on the place where neural tube is created, and that cells migrate across the body on the different parts of the body, and that cells are responsible for development of different body parts, organic system, and so on. So, and including those parts which are involved in domestication syndrome. To summarize, the mechanisms behind domestication syndrome are therefore relatively well explained already. So there are two main components: neuroendocrine system, HPA axis, thyroid glands, and so on, and neural crest cells. So that's two. But, okay, that are explanation of the mechanisms. What about the cause? Is there maybe any cause, initial cause, to trigger these changes, these mechanisms? So, and the question here is, could it be parasites? And now we are, that's it, what it is about the parasite-mediated domestication hypothesis. So let's say the main premise of, this hypothesis mainly derive, or the main premise is that parasites somehow mediated domestication, at least initial phases, so that they initiate some changes that leads to domestication process, let's say. There are some premises that some things which we have to be aware on in the context of this hypothesis, and the first is that parasites, as you probably know, are known as evolutionary engineers. So they are shaping evolutionary process. They are across all over the animal kingdom, shaping some processes of evolution. So they are so much involved in evolution of the animals. And parasites, directly or indirectly, are involved in literally all the main processes that underlie domestication syndrome, which I mentioned before. So neuroendocrine system, directly by shaping the behavior. You know that parasites usually shape animal behavior in a way to increase the probability to spread across the population. So there could be also an indirect impact of the parasites through the miRNA. You're probably mostly genetics here. I'm not genetic, but anyway, miRNA that are small, highly conservative, non-coding molecules that dictate or control expression of genes. So, and as we know, parasites could change the profile of miRNA in the animal, in the host animal. So that's one thing. The second is neural crest cells. So they can indirectly also affect neural crest development through miRNA. So that's theoretically. And the third thing is disruptive selection. It is known that parasites cause increase in phenotypic and genetic variability in the host population. So we could say also disruptive or destabilizing selection, although concepts are a bit different. Here, as a point of interest, it's a table with the miRNAs, families and types, which are interestingly involved both in neural crest development and are shaped. The profile of that miRNAs is changed by the parasites infection. So here is some overlay of that thing. So in general, PMD, our hypothesis predicts that the frequency of domestication syndrome traits in the wild population increase with decreasing genetic resistance to parasites or with increasing parasite load. So, and it proposes that the domestication syndrome could be genetically linked to genes related to resistance, tolerance to parasites, or the role of miRNA in the process of epigenetic inheritance, including transgenerational inheritance of stress pathology. Okay, there are several ways to test this hypothesis. First is comparative approach, and the second is experimental approach. The second one we will discuss in short at the end of this presentation. About the comparative approach, we can compare simply like genetic resistance or parasite load in, let's say, wild population and domestic population in the same environment. So the population that have similar chance to encounter parasite fauna at that area. So, and we simply check the parasite load and compare them. And or to compare fully wild animals with wild animals showing some signs of domestication syndrome and so on. And here we followed a systematic comparative approach in this our study, and we simply compared the parasite load in wild boar and free-ranging domestic pigs. So, which we had a pool of wild boars and free-ranging domestic pigs. It means domestic pigs that were on pasture outside. So that population were from the close geographic area, which was the important precondition of methodological precondition. So here is a picture of study sites. So we had two main study sites, one in Slovenia, this above, and below in Croatia. So both in each country we got two populations, wild and domestic, which were relatively close, up to 20 kilometers. So 20 kilometers is not much for wild boar, which is so explorative, let's say. And of course, from that animals we collect simply fresh feces from rectum in wild boar, hunted wild boar, and right after defecation in domestic animals, and did some diagnostics, simple diagnostic for parasites. So what we have found, actually, in general. And these are preliminary results. So we found highest parasite richness in domestic animals, which correspond to our hypothesis, which is in line with our expectations. So we found. In this case, two taxons of parasites more in domestic animals. So we further, of course, analyzed also a particular parasite, because not necessarily that the general parasite load is involved in this process, but could be also some particular parasites. And here the most interesting, that there were some parasites found only in domestic, only in wild, but there were also 3 found in both. And 2 out of 3 were significantly higher in domestic pigs, which is also consistent with our hypothesis. Okay, so let's conclude and discuss this a bit. So in general, of course, our results are consistent with our hypothesis. Although, of course, it is a preliminary study, we cannot give any definitive answers. But anyway, there are some indications in favor of this hypothesis. But of course, to study and compare parasite load is a bit problematic. We will tell a bit about this. So why? As I said, parasite load could be a bit problematic because there are 2 aspects to be aware of. Possible artificial selection of resistance in domestic animals. So we can, in case that domestic animals were selected for parasite resistance, so of course we can get results which are misleading. But anyway, in the pigs, in breeds we dealt with, there were not selection on parasite resistance. So, and the other thing is opposite situation, relaxed selection, when you eliminate vector, selective pressure, in this case parasites. So domestic animals didn't have that much opportunity to encounter parasites like wild one, and this could shape or influence the evolution of parasite resistance. So this could be also a case. But anyway, in the pig, domestication does not appear to act as a bottleneck limiting the diversity of parasite resistance genes. So, besides comparative approach, of course, the most reliable way would be experimental approach, like the same proto-domestication experiment as Beliao-Fox experiment or Trapezio-Mink experiment. would be the best. But we wouldn't select them for tameness, but simply we would have experimental populations, expose them to the parasites experimentally, select them against parasite resistance, so more infested, less resistant animals, and record frequency of domestication syndrome traits over generations. So we expect them to increase, simply. But of course, as experimental approach is problematic and above all time-consuming, there would be an optimal alternative for this. So to do, because you have here like two perfect populations, let's say in the context of the domestication process, foxes and minks. So the simple approach would be like a first step, just to test dead populations and different types of animals, aggressive, domestic, tame, and compare them, compare their parasite load, compare their genetic resistance against parasites, and even maybe miRNA profile of that animal. So specific families which I showed before. Okay, that was really short. I hope I'm still in time. Thank you very much. Spasibo, hvala.
Thank you for a very interesting presentation.
Here are some references.
Question, please.
Вопроси может быть тоже на русском. Но я постараюсь ответить.
Okay, maybe English is still easy.
Okay.
First, I give you one comment and one question. So the comment is, we are working with Professor Trapezov about mink and have very fancy single-cell profiling of a brain. And to our surprise, one of the very clear difference between tamed and kind of aggressive minks is evidence of some kind of brain inflammation. So we are very curious, and maybe one of the idea that they just infected with something that's kind of through parasites affecting the tameness or aggressive behavior. So it's kind of in favor of that. But kind of critical comment or maybe question that I have. So in my sense, this kind of experiment rather shows that domestic animals would have kind of suppressed immune system. And then you let them go, they go to the field and they immediately infested with the parasites, and that's what we see. I think you commented a bit in your talk about this, but in my sense, this is one thing. And a small question I have, we have a request from industry about pigs. They are very kind of curious if there is some kind of subpopulation always in the industry, in the farms, that shows the extreme aggression, right? So they bite other pigs, so it's a big damage to the industry. So they ask us to kind of try to find a cause, in effect. So do you think the kind of presence or lack of kind of parasite infection can be considered as one of the factors?
Yeah, also, as I said, yes, there can be any definitive answer can be done, so maybe it could be. And about the first thing you mentioned, yes, of course, there is many aspects to be aware of, so that can mislead us in interpretation. And not necessarily, of course, the suppressed immune response of animals is should also be under the question. Of course, I agree. Yes, and it's true that parasites in many cases make animals simply sick only. But there are some parasites which are really sophisticated within the animals. We even don't know that they are there and so on. And also, it is not necessary that parasites mediate all the process of domestication. Maybe only, as I mentioned, it is maybe only an initial stage. So there is a lot of questions, a lot of aspects, and thank you for the comment and about the brain inflammation. It's also very interesting. So I don't know if I answer your question, but as I said, there is no complete answer. There is still so much more questions than answers, and maybe the idea is completely wrong.